Good morning. I would like to welcome everyone to the session entitled Immune Models to Predict Toxicity and Efficacy of Gene Therapies. It's organized by the Immune Responses to Gene and Cell Therapy Committee. I would like to welcome Dr. Antonia Folenzi, and my name is Paweł Murański. The first speaker is Dr. Paola Grandi from University of Pittsburgh School of Medicine. The title of her talk is Developing Oncolytic Therapies for Brain Tumors.
Well, it's working. Thank you. Well, thank you for giving me the opportunity to present our data. And all this data has been generated at the University of Pittsburgh, but I'm now, and I'm disclosing this information, I'm now a scientific founder, and I am a director of Immunology Virology program on Chorus. So, the disease we are tackling is GBM, so adult glioblastoma. It's one of the most common and aggressive neoplastic malignancy. The incidence, as you can see, is 3 patients have a 100,000 person and 22,000 are the new diagnosed per year and 13,000 the death per year. The 5-year survival rate is less than 5%. Standard of care really doesn't work at all. Neither neurosurgery, chemotherapy, radiotherapy, and temozolomide that was approved almost 10 years ago in combination with radiotherapy can improve the expectancy of life of just 2 months. So what are the major issues with this kind of tumor is infiltrating the entire brain, and it's really impossible to go for a surgical resection. Is genetically and phenotypically really heterogeneous. It's always recurring. Even after surgery, after treatment, is always recurring. And the prognosis depends on the type and the grade of glioma. So it's been done, this study has been done at the MGH, in which they took a single biopsy of the patient and they did a single cell RNA analysis. So these GBM are generally classified based on the mRNA profile on mesenchymal, classical, neuronal, proneuronal. However, you take a single biopsy, you analyze cell by cells, and you're going to realize that in a single biopsy there are different kinds of subtype of actually this tumor, and there are cells in which we have a mixture profile. So again, we are facing an hybrid state, and we are really facing a really heterogeneous situation. And these are 4 different patients. 4 different patients who have the same disease, GBM. Each one of them has a different location, completely different symptoms, from behavioral issues, vision, language, or issue on walking and moving. So we really need to find a therapy that can target the heterogeneity, can adapt with the tumor itself, as you can see, is really different, and that does not depend on the complex mutation of the tumor. These tumors are also driven by different key mutation. So the field for years and years thought that we could develop this oncolytic vector, and this oncolytic vector will go and will kill all these cancer cells. Well, we actually learned that it's totally unlikely to expect a virus to go and kill all these cancer cells, especially when we have such an invasive tumor. However, what we also learned is that oncolytic vector therapy, oncolytic virus have the ability to kill these cancer cells and to recruit an immune response that in turn is going to recognize the tumor-infected cells. So how the virus does that? By killing the cancer cells, he really elicits an immunogenic cell death, all these dangerous signals such as HMGB1 and ATP and DNA and RNA that get released. So are these actually dying cells that they work as the really strong immune stimulus? So when we think about oncolytic vector, we really need to think about the fact that their therapeutic profile, their efficacy rely on 2 different aspects. 1, we need to have a virus that robustly replicates in cancer cells. 2, we need to have a virus that kill efficiently, trigger an immune response, and as a consequence, we need to have a virus that can replicate even in the presence of an immune response. So this is our wish list for everybody who's working on oncolytic vector. We want to have a virus that is not pathogenic to the normal tissue. We want to have a virus that can go and infect an heterogeneous tumor, different kind of tumor cells. Has to be able to efficiently kill them, has to be really stable, and also upon, of course, this replication inside the tumor itself. Must be able to replicate even in the presence of neutralizing antibodies, so we can actually inject the patient multiple times. Must be able to turn that anti-inflammatory tumor microenvironment in a pro-inflammatory tumor microenvironment. Must be able also to recruit an immune response. Must be also have enough room so that we can arm the virus with our gene of interest. And of course, all of this is what we all want, but what we really have is not that there yet, but we're working on it. So what do we do and where we are going? So what we knew is that there are several barriers that can really block the tumor therapy in patients. One has been the poor virus replication. A lot of viruses don't replicate as efficiently as we want. And as I said, if they don't kill the cells immediately and robustly, then it's really hard to think they're going to trigger an immune response. There is a limited viral distribution. For most of the solid tumor, we're talking about intratumoral injection. The virus doesn't spread efficiently. As a consequence, we have a really limited amount of cells that get killed by the virus. Most of the oncolytic vector, they don't have the ability to change this tumor microenvironment, and as a consequence, they lack the ability to recruit an immune response. So what can we do? You must know that the field worked for years and years and years to make this virus as safe as possible. And as a consequence, some of the key genes of the virus has been deleted in order to ensure the safety. But what we really need to figure out is how to make a virus really safe and still retain all this viral function that has been built in to really take over the infected cells. But if you think in that way, then we need to find a way to make this virus really selective for cancer cells, being able then to infect and replicate on in cancer cells. We also need to make a virus that can spread efficiently, and as I said, in our dream list is also a virus that is able to trigger a strong immune response. So the vector that, HSV-based vector that have reached the clinic so far, they're all characterized by the mutation in this gene called gamma 34.5 minus. That deletion was actually in the first vector that Dr. Martusa made, and the first vector went in the trial, and the rationale was that this gene is important for the neurovirulence, so if you remove it, the vector is safe. And the second mutation that most of the virus has is a deletion in ICP6, who's encoding for the ribonucleotide reductase. So without this gene, the virus can replicate only in active dividing cells. Since active dividing cells, they express the large subunity of the ribonucleotide reductase. However, we also learned that gamma 34.5 is a really important gene. What does it do? So if you think this is your virus, and your virus is infecting the cells, there are several sensors inside of the cells that are able to immediately try to block vector replication. There are DNA sensor. There are the toll-like receptors that are getting activated. And overall, the cells react by forming this big complex that is led by TBK1. When this complex form, IRF3 gets phosphorylated, and all the interferon responsive genes are getting expressed. So the virus comes with this gene, the gamma 34.5, that actually block TBK1. By blocking TBK1, we don't have the expression of all the interferon responsive gene. Furthermore, the virus, as you can see, upon infection also start to produce double-strand RNA, of course, which in turn activate the PKR. And this PKR induce the phosphorylation of IF2 alpha and block the translation of the viral protein. Again, the virus comes with a gamma 34.5, which can dephosphorylate PKR, and as a consequence, allow the translation of the protein to occur. So in the Oncovax virus, the virus that went on a clinical trial, gamma 34.5 has been removed. However, the US11 gene that has a similar function that gamma 34.5 has been put under immediate early promoter to ensure a really robust expression so that this process can still occur, meaning the virus can still hijack the cells and allow protein translation. Still, US11 does not compensate for this set of pathway. And why this is so important? It's not just important because this is an interferon responsive gene that can block vector replication, but because one of these key genes is actually IDO. And I don't know how familiar you are, but IDO is one of the genes that also the cancer cells are producing to induce the Treg and block CD4 and CD8. How do they do that? IDO is this enzyme that takes these key amino acid, the tryptophan, who is an essential amino acid, and metabolize the tryptophan in kynurenine and quinonilate. So there are two things here that are happening. Tryptophan is an essential amino acid. If you have a virus that replicates really efficiently, it's going to take this tryptophan to build all the structural protein. But furthermore, this vector not just sequesters tryptophan and shut down the enzymatic activity of IDO. Furthermore, what this virus does by blocking the interferon responsive genes is also blocking the expression of these key protein. As a consequence, block the activation of Treg and improve CD4 and CD8. So this is why it's so important to have gamma 34.5 in the virus. So we reason that if you want to keep all these genes, if you want to have a gamma 34.5 virus, we need to make it safe. So the first thing we did, and that was Dr. Rucida and Joe Glorioso's lab, and we retargeted the virus to the EGFR receptor. So normally this virus has a lot of glycoprotein. GD is the key glycoprotein that recognizes two receptors, HPM and Nectin-1, and upon the binding, there is the fusion that is mediated by the other two glycoprotein. So what we simply did was engineering the glycoprotein D so that instead of binding to its cognate receptor, now this glycoprotein D can bind to the EGFR since we clone a single chain antibodies for the human EGFR, recognizing the human GFR and the human EGFR-B3. And I'm not going to go through all the data, but what I want to show you is that if we inject this virus into the brain of the animals, what we observe is that, of course, we have no toxicity. This virus is really specific for the human EGFR and EGFR-B3. But when we test this virus in a xenograft model in which we injected into the intracranially orthotopically these human GBM30 primary glioma lines, we observe that the animal that receive the PBS, the vehicle, they all die by day 35, while the animals that receive the vector that is retargeted to the EGFR receptor, now they can, we were observing like a 60-70% long-term survival. Now, yes, we retargeted the virus to cells expressing EGFR. These still is not like a CAR T cells, it's a different concept, and it's not like using antibodies against EGFR. And the reason is, here we are taking advantage of something that the cells is expressing, so the virus is killing them, and we actually release all these tumor antigen. As a consequence, we do not quite pre-select a single antigen in order to create an immune response against that specific antigen. When you think about this oncolytic vector, you have to think about this is like almost your ultimate personalized vaccine system, because when you kill them, you really release several tumor antigen. What you're shooting for is an immune response against most of them. So, great, we could retarget the virus to EGFR, but we also knew that EGFR is not expressed just in cancer cells. So what can we do to ensure the replication of the virus just to cancer cells? In virology, in general, most of the viruses have their genome divided in two different groups. Genes that are essential and genes that are not essential. Without the essential genes, the virus does not replicate. Without the non-essential genes, the virus can still replicate in vitro. In vivo, it depends, because again, if this not essential gene is involved in the immune response, then when you put it in vivo system, that can be still really important. Still, there are this gene that is ICP4, that is one of the essential genes. So if we abrogate the expression of ICP4, the virus cannot replicate anymore. Furthermore, ICP4, that is the gene I'm going to talk about right now, is not just essential, it's also an immediate early gene. So viruses are pretty smart on the way they replicate, and it's really well organized. So we have immediate early and late gene in a really chronological order. So if you block the expression of an immediate early essential gene, you fully abrogate the expression of all the rest of the protein, including DNA replication, include the expression of all the early and late gene. So we reason that if we want to ensure the safety of the virus, we really need to abrogate the expression of ICP4. And what we did, we took advantage of the differential expression of microRNA. So healthy tissue, like neurons, have high level of expression of miR-124. When I talk about high level expression, I mean 8,000-fold to 0. miR-124 is the key microRNA that is expressed in neuronal cells and turn on all the neuronal genes, and as a consequence is the key microRNA that control even the cell cycle. If microRNA 124 is expressed, the cells cannot divide. So its expression is totally incompatible with a tumor phenotype. So we did something really simple. We took our ICP4 gene in the virus and we simply add the target sequence of miR-124. So in the presence of miR-124, like in neuronal cells, miR-124 bind to the target sequence and block the expression of ICP4. At the opposite, in healthy tissue, in cancer cells where miR-124 is not expressed, the vector can replicate as well as the wild-type virus. So this is exactly where we're going. We wanted to have a virus that can infect and replicate efficiently in cancer cells, and which, upon infection of neurons, the expression and replication would be completely abrogated. And we knew there were several microRNA that they are exquisitely expressed only in neurons and virtually totally absent in cancer cells. As a proof of concept, what we did, we took our virus that is otherwise a wild-type virus and has just the miR-124 target sequence controlling the ICP4. And we did a straight injection into the brain of the animals. As I said, this is practically a wild-type virus. It's just this 100 base pair, the 3' UTR of ICP4. And we observed the total non-replication of this virus in the brain of the animals. And now we already repeat those experiments using 36 animals, really high dose, and we know that this system really works. What we did, as I said, as a control, we used the wild-type virus. And these are just the staining of how the brain of these animals look like. So we couldn't depict any viral antigen, suggesting that really the virus cannot replicate, and we did not observe any neurological symptom out of these animals. So what we did next, we combined our two approaches. We made a vector that was the first vector that I described before was retargeted to EGFR, and then we combined the miR-124 approach with the EGFR approach, and we repeat the experiment using the xenograft model with the GBM30 and observed that by adding the target sequence for miR-124 in the virus, this virus was absolutely not impaired in efficiency of replication and killing these cancer cells. So now, based on this data, we don't even know if we're going to keep using the retargeted virus because the miR-124 control is so strong that we can probably just use the GD wild-type virus to infect our cells. But we are still facing a big problem. When we're talking about this tumor have a robust extracellular matrix. And what we really want, as I said, is having a virus that can improve, that can replicate and spread really efficiently in the extracellular matrix. So I did this experiment quite a bit ago. We were using this animal model. This is a window. You can open the window, implant the tumor, close the window. and check in vivo what is happening inside of the tumor. And this is actually, I don't know if you can see it nicely, but this is actually an in vivo imaging about what is going on inside of a tumor. The red thing here is a pseudo color for the collagen, so the protein that is part of the extracellular matrix, and this is where the virus was injected. Again, I don't know if you can see it nicely, but what we consistently observed was, doesn't matter if we're using the Harvard pump syringe and so on, every time we were injecting the virus, we were having an area of the tumor that was obviously highly infected by the virus. We can see all these green particles, but we could also observe that beyond this point, the virus was not able to move and spread. And so if you do a pixel analysis, what you're observing is that where you have the extracellular matrix, you don't have virion particles, and the opposite, where you have the virion particles is where you mechanically destroy the extracellular matrix. Now you don't see the collagen. We, as a control, use a dextrin, so it's a really little molecule, and observe that, as expected, at the site of injection, we still have this mechanical breakage of the extracellular matrix, so the dextrin is in one part where you don't see the collagen, but on the other side, by going just borderline, we could see that dextrin could go where the extracellular matrix was. So the point was, why this is happening? Is this virus just because this envelope virus is sticking to the extracellular matrix, or is it because the virus is big? And this is a little bit more about how the situation looks inside of a tumor. This black spot that we call the honeycomb structure are actually your cells. And you can see the virus really getting stuck, attached to the protein outside of the cells. So going down a second, this is 250 micron. If you see here, if we treat the tumor with collagenase together with the virus, so now we can see and observe a really nice spreading throughout the area of our virion particles. So in order to understand if it was because the virus is big or the virus was sticky, we use microsphere. The size of this microsphere was 200 nanometer, so fully resembled the size of the tumor, and observed that the microsphere were enabled to spread as well as the virus, suggesting that it's a matter of size, it's not a matter of the virus being sticky. So how can we fix this problem? So we decided to arm the vector with MMP9, a metalloprotease, and ask the question, can a virus that expresses MMP9 now infect and replicate and spread more efficiently than the parental virus? One thing you need to know about our vector is also we have 20 kb of space, and so we can easily kick in and out a different transgene of interest. And when we use the MMP9 and we test it in a spheroid model, we observe that MMP9 nicely improve the spreading inside of the neurosphere, that they are GBM-derived primary neurosphere. Of course, this is just an in vitro experiment. So then we took it to the next level. We use again the GBM line, the GBM30. And in order to depict the therapeutic profile of the virus arm with MMP9, we really need to use an input of viral that it was just 10 to the 4 PFU. So if we were going back to the experiment we did before, we had to use 10 to the 6 to see a long-term survival in our control vector, and in this case is the retargeted MIR124. But if we express MMP9, we can achieve 70% survival by just injecting intratumorally a total of 10,000 million particles. But what is most of these experiments are kind of meaningless, and this is what everybody are doing in the lab. You inject the cells, and after 8 days, 7 days, when you don't even have a well-structured extracellular matrix, you go and you inject your virus, right? So we try to mimic really what happened in patient. And we repeat the experiment by injecting the tumor cells and then wait for the tumor to be formed up to almost to the extreme, meaning these animals were symptomatic. The tumor was almost a third of the entire brain tumor, or the entire brain of these animals. And after we normalize the animal based on the volume of the tumor, then we inject and treat the animal either with the control, the vehicle, the PBS, with our control vector that is just a retargeted miR-124, or with the virus expressing MMP9. So this is actually the differential in tumor volume upon treatment. As you can see, the PBS in 13 days is like after we inject the PBS, the tumor really pushed almost toward the other hemisphere. What we observed was the control vector. Again, sorry, this is a xenograft model, so we don't have an immune response. And what we are depicting here is just the pure oncolytic activity of the virus. We can observe that our vector can definitely replicate this better than the PBS and can slow the growth of the tumor. What it was kind of really interesting was observing that if the virus expresses MMP9, again, we are not curing this tumor, but we fully stabilize the disease, suggesting that MMP9 really improved the therapeutic profile of the virus. So, but it's really important for all of us to understand, as I said, besides the oncolytic activity of the virus, to which extent the virus can trigger an immune response. And sadly, in our field, when we're talking about syngeneic model, the syngeneic model are based on standard cell line that has been used since forever. Probably the one that most of us are using are the GL261. And murine lines that are quite refractory to infection with human virus. Doesn't matter if you're using an adenovirus, a measles virus, an HSV virus. All these murine cells are more refractory, so they partially mimic what you would see in a patient. On the other hand, they are not just more refractory, but they are definitely more immunogenic. So we know that these GBM tumor actually are definitely not immunogenic, but what we are working on is with animal models that are highly immunogenic. And this is just to show you how this model, especially when you're using GL261, does not fully translate to what we will see in the clinic. And was a beautiful paper, and then the data were presented also at the ASCO meeting, in which the animal were injected intracranially with the GL261, and the animal received either the IgG control, the CTLA4, the PD-1, or the combo. As you can see, this is PD-1 by itself. They have 50% long-term survival. You go for the combo and you reach 70% long-term survival. And what we learned just last week is that actually in clinical trial, none of the patients really respond to the anti-PD-1. So this is a huge limitation. We cannot even challenge our animal with GL261 because, as I said, they are so immunogenic that it doesn't matter what you're doing, you're going to have beautiful data because you're going to trigger an immune response against the cells. Furthermore, though, going back to how resistant these cells are, these are several HSV-based vectors. They're just an example. But it's also telling you that in order to kill 50% of your monolayer in vitro, you need to use, for most of these vectors, more than 1,000 PFU per cells. And this is not what is happening in any human cell line. So what can we do? There is any animal model that we can use and actually more or less resemble closely what is happening in the clinic. Yes, there are the AirCast CVGBM model. So this model, as well as sleeping beauty system, are systems in which you use genetically modified animals in such a way that you can induce the formation of the tumor by injecting either shRNA or viruses that they can shut down or induce the expression of some of the either oncosuppressor gene or oncogene. So in this case, we're using the AirCast model. This is a genetically modified virus, and that it comes in two different versions. In one, there are the nesting positive cells that they express the TV receptor for the avian virus, or the GFAP positive cells, so the astrocyte, that they express the receptor for the avian virus. So what happens is you can inject this avian virus that they're carrying by cells, and this virus can contain either SGLT53, PDGF1, IDH mutant, based on how you want your tumor, which kind of genetic signature you want your tumor to have. So this is just an example when you shut down an F1 and a P53, and this tumor actually fully resembles a human tumor in terms of heterogeneity, because they're not that just chronogenic, and in terms of infiltration. And also in terms of how this animal respond to the standard of treatment. So after you inject this animal, most of these animals start to develop a tumor. By 21 days, they become symptomatic. It's a big tumor, and they normally succumb to the disease in 30 days. You treat the animal with temozolomide, and in your best scenario, you improve the expectancy of life of this animal like 5 to 7 days. When you add radiotherapy, now you improve the life of these animals of 2-3 weeks. So this animal really is as close as you can get to the human. Who has this animal model is Eric Holland in Seattle. Before it was a Sloan Kettering Center, so also the research that has been done toward the CSF1R, and then, of course, the translation in the clinic has been all based in using this animal model. So one thing we did, we took the cells from this animal, the cancer cells, and then we asked the question, can our virus actually efficiently infect and replicate in the cells? And the answer has been yes. And another important thing about this animal model is if you do the full characterization of the tumor microenvironment and you compare what you have in these mice, in this case it's being compared between a tumor that is IDH mutant versus a tumor that is IDH wild type, then you will see that this signature fully resemble what you see in the biopsy of the patient. So I have only 4 minutes left, so I'm going to go fast now. Sorry. Again, this is if you compare 1-to-1, this is what you have in wild type, IDH wild type patient, that are mainly high-grade glioma patient. This is what you have in low-grade glioma patient. And as you can see, even the immunological signature between these two tumors is quite different, with a huge prevalence of NK cells, and this is probably what you need to fight when you have a wild type, IDH wild type tumor, versus a really huge, strong prevalence of macrophages and MDSC when you're facing low-grade tumor. And so what we did using this system, we injected our virus and we asked the question, can our virus actually trigger an immune response? And these are just preliminary data. But what I can see, we heat inactivated the virus. So the virus, even without any kind of replication, can induce infiltration of lymphocyte. This is our virus expressing MMP9. And when we combine our virus with immunocheckpoint inhibitor, now we were able to observe a really strong infiltration of T cells. And in a really similar way, this is by looking at the presence of CD8. So, as I said, we are in the process now to fully characterize also the therapeutic profile of the vector. I spoke a lot about our vector. However, in the clinic, there are so many other viruses who have great potential. Yesterday, we heard about the measles virus. On the next door, they're talking about actually the TOCA511. Really great data on the adenoviral vector. And I'm going to tell you a little bit more about actually these 3 trials going on right now. So this is Dynatrax. They developed this oncolytic vector, and they use a different system to ensure the safety. So what they did, they make a deletion in the delta 24, and to improve the efficacy, they actually engineer one of the fibers so that this virus can efficiently infect and replicate exquisitely in cancer cells. Again, they also did the full analysis of the tumor microenvironment, and they show that the virus can efficiently replicate, kill the cancer cells, spread really efficiently, and can induce a strong immune response. Furthermore, they have the data that everybody wants to have, right? They have patients that respond to their treatment. The major issue that we are facing is what is the signature, why some patients respond and some patients don't respond. Nevertheless, these data are really strong proof of principle that if you have a strong oncolytic virus that can replicate efficiently, this virus can trigger an immune response, which in turn kill the cancer cells. And because as you can see, upon resection, you still have a big tumor. And as a matter of fact, you ensure not just a long-term survival of the patient, but also a great quality of life of the patients. So now, Dynatrace already treated more than 125 patients, and then next June we're going to have more data regarding the long-term survival. In a similar way, TOCA511 is not an oncolytic vector, it's a virus that's integrating into the genome of the cancer cells, can exquisitely then replicate together with the cancer cells, and is expressing a prodrug so that is the cytosine deaminase. So when then you provide to the patient the FC, that gets transformed in a 5-fluorouracil, which in turn kill the cancer cells. As well as for the Dynatrace trial, also TOGA511 has really good promising data. We would love to see even more positive data, but nevertheless, there is an improvement in expectancy of life of these patients of at least 2 months, and the quality of life is so much better than what it was on the standard of therapy. So where are we? So we developed this oncolytic vector. We're going to do the full characterization, and as I said, what we really all want to is let's bring clinic and let's try to be able to treat this patient and improve their quality of life. And all of this work comes from primarily something that Joe Glorioso started, great HSV virologist, gene therapy, former president of the society, to actually create this huge group of people, and it took them 20 years to get where we are. We always closely collaborate with Nino Chiocca, that has been instrumental in helping us really bridge from just the bench to the translational side of it. And this team, together with, they worked together with us, and most of the research was also sponsored by the NIH. Nevertheless, Eric Holland is what is taking us to the next level. And as I said, now I joined this company called Oncorus based on our technology. The team is outstanding, and that what is going to allow them to go from walking to running to the clinic. So thank you so much.
Thank you very much. We have time for only one quick question.
Come to the microphone, please.
As you're using MMP to degrade the extracellular matrix in tumor, I was wondering if there were any safety concerns, as MMP has been implicated in also metastasis.
Yeah, so a really good point. We did all the experiments, so we overexpress the MMP in vitro and MP9 in vitro, and we look if that could improve the infiltration and invasion of the cancer cells. We didn't observe that. We were really puzzling about what we were seeing in vivo. Got in touch with a molecular biologist, structural biology down to MIT. Oddly, they were observing exactly the same thing. So when you overexpress only one MMP, somehow this MMP that really works therapeutically speaking, and by overexpressing one, you practically do not allow this complex to occur, so you do not improve in the invasion and of the cancer cells. On the other hand, you still can digest efficiently the extracellular matrix. So we have that set of data, and we have all the data in which we can inject this vector harm with MMP9 with really high level concentration to the brain of the animal, and observe none whatsoever toxicity or change when we are not in a context of a cancer. So
thank you very much.
Thank you. Okay, thank you very much, Paola. Sorry if we don't for more questions. We have to go on. Now, it's a pleasure to invite Leslie Keane, that will give a talk on modeling immunotherapies in non-human primates. Thank you.
Thanks so much for the invitation to give a talk today. Here are my disclosures. So today I'm going to talk to you about the work of my lab, where we spend a lot of our effort in building complex non-human primate models. And here I'm going to tell you about the mechanistic insights that we've learned, both about graft versus host disease after allogeneic transplant, and then a new model that we've developed to study CAR-T neurotoxicity. So as we all know, relapsed leukemia presents a difficult problem with currently really imperfect solutions. And I think this is illustrated very nicely in this slide from the Children's Oncology Group in 2008, where you see that the survival, even in pediatric ALL, which is known to be sort of some of the best survival, after relapse is really dismal, even for the lowest risk patients, and almost, impossible to cure for the high risk patients. Currently, with those patients, allogeneic stem cell transplant is still the best chance for cure, but we really have a long way to go here. In pediatric ALL, we collect data, in hematopoietic stem cell transplant, we collect data on every patient that is transplanted in the country and in many countries around the world. So this is data from the CIBMTR, which shows you that the number of transplants that we do each year has steadily increased, again, because hematopoietic stem cell transplant for cancers really does represent the state of the art right now. However, despite the fact that so many patients are being transplanted, there are many deaths after transplant. And if you look at the cause of deaths, really, again, it's very clear that relapse of the primary disease is critical. And I'm going to get to sort of our approach to solving this problem later on in the talk. But one of the most devastating things for a transplanter is when you have a patient for whom you've cured their leukemia and they die instead of a toxicity of transplant, the most devastating of which is graft versus host disease. And my lab has been directed towards understanding and correcting this complication for many years. So the question that we approach in my lab is, how can we identify the critical mechanisms controlling graft versus host disease after transplant and develop novel therapeutics? And it's this work that has led to some new clinical trials and also to our work in CAR T cells. So we chose many years ago to base our studies on a systems-based mechanistic platform in non-human primates and in patients. So when you think about creating models in non-human primates, you really have to reach two bars. It has to be better than mouse studies, so you have to be able to answer questions in the non-human primates that you really can't answer in mice. And you also have to be better than human studies. You have to be able to ask deeper mechanistic questions than we can easily ask in clinical trials. And in fact, for many studies, non-human primates represent a really ideal model. One of the most important things when we're modeling cellular therapies, either transplant or CAR T cells, is that the immune system of the primates really, of all the models, most closely resembles patients. And because of that, really, and this is really a key, many of the novel biological therapies are cross-reactive in primates and not in other models. And then again, as that value added compared to human studies, we're able to do intensive multi-organ longitudinal analysis in primates that are more difficult to do in the context of trials. And so the central strategy for my lab is to do detailed studies in non-human primates for rapid translation to patient care. And so to do this, we had to build the model. So before we started working, there were many models of autologous transplant in primates, but there wasn't a model of allogeneic transplant and graft versus host disease. So our transplant paradigm tries to recapitulate human transplant as much as possible. So we have the non-human primate recipient here, but as I'll show you, everything we do to the non-human primate is really based on what we do in clinical transplantation. And so what we've been able to create over the years is a system where we can essentially dial in the degree of transplant complexity or simplicity or preventative therapies or treatment therapies, just like we do in patients. So we've now MHC matched several colonies around the country, so we can dial in the degree of MHC disparity. We can have matched donors, we can have mismatched donors. We can also do very important autologous transplant controls. We can dial in the degree of intensity of conditioning, myeloablative conditioning, reduced intensity conditioning. We use the exact same reagents as we use in patients. And for the majority of the studies I'll show you today, we've used a TBI-based conditioning regimen. And then very importantly, as we want to study GVHD, we can then dial in different GVHD prevention and therapy approaches. So in the work I'll show you today, I'll show you that we've used both standard of care prophylaxis using rapamycin or sirolimus, tacmethotrexate, and also some novel preventive strategies using CTLA-4 Ig or abatacept and sirolimus. So we can study all of these prevention strategies. We can do a lot of clinical analysis, and I'll show you that. But really the strength of the work that you can do in non-human primates is you can do a lot of very intensive analysis of the immune response to transplant. So what we've taken as one of our major paradigms is to sort T cells during and after transplant and look at them in a very detailed manner, both by flow cytometry and by transcriptomic analysis. And again, as I said, one of the strengths of this model is we not only can look at the peripheral blood, but we can also look at the GVHD target organs. So just to remind us all what we are combating when we're combating GVHD after transplant, this is a multi-tissue disease that really is devastating both for quality of life and also for its high rate of mortality when the disease is severest. You see GVHD of the skin that in its most severe form resembles a grade 3 or grade 4 burn. You see GVHD of the liver resulting in jaundice and lymphocyte infiltration into the portal triad area. And then you see GVHD of the GI tract, currently the most deadly form of GVHD, where you can see both gross ulceration of the GI tract and again T cell infiltration into the GI tract. And so just as we can monitor GVHD clinically in humans, we can also do this in non-human primates. So one of the strengths of the model is that we can actually look at all the same characteristics in non-human primates as we do in humans. So we have a clinical score just as in the patients where we score rash, bilirubin, and diarrhea. And here's just a list of many different primate cohorts that we've studied in the model so far and how they do in terms of their GVHD clinical score. So you can see here that if we don't give any therapy, so if we just look at the natural history of the disease, these animals develop very severe GVHD very quickly, and then we've actually been able to identify and have published some really novel combinations that really can totally control GVHD. Now, because our model is a natural history model, it means that when we get GVHD, we do not treat it, right? So we actually look at what happens to the GVHD that develops in each of these either prevention or treatment paradigms. And because of that, the severity of the clinical GVHD reads out very nicely as survival. So we can actually use survival as a surrogate marker. Now I'm going to talk to you a lot about our sort of complex immune analysis that we can do, but this model also lets us do sort of the traditional analysis that we do in patients. So I'll show you first some histopathology. And so what we published many years ago now is that the histopath of the primate GVHD model really closely recapitulates human pathology. We see pathology in the skin, nice example of T cells infiltrating the liver here, and the GI tract. One of the things that the non-human primate model has allowed us to do is look a little bit into other organs as well. And in fact, this really prompted our CAR T cell model that I'll show you later. So as we were looking at our animals who developed GVHD, one of the things that really struck us, and we actually commented on in our first paper about the model, was that these animals exhibited neurologic complications. So they were neurologically not normal. They often laid down their cage, they were ataxic, etc. So this prompted us to wonder whether the brain, as well as these sort of canonical target organs, could also be target organs for GVHD. And in fact, just at the time that we were publishing our work, and this was published in Blood in 2014, Marcel van den Brink and his group at Sloan Kettering also published a very similar observation in the mouse, where they not only saw T cell infiltration into the brain, but also they could do some nice behavioral studies in their mice with GVHD. And so what we saw in these animals with GVHD, very distinct from normal animals, is that you really get a lot of T cell infiltration. And this is really interesting because those of us who take care of patients with GVHD know that there's some cognitive problems there, but until the Marcel's paper and our paper really hadn't been able to quantify it. So you can see that these T cells, these highly activated T cells in GVHD enter the brain. And that's going to be very important when you think about CAR T cells as well. So I told you that we can look at things clinically and we can look at things histopathologically, but I think the greatest impact that our work has had over recent years is really to take a systems approach to this and look at the transcriptomics of GVHD. So what I showed you before was sort of the splitters look at GVHD. So every different prevention paradigm that I showed you, we look at their score, we look at how they do. But you can actually kind of be a lumper in GVHD as well, and here I've sort of lumped prevention paradigms that result in very rapid death and very severe GVHD, and those that for a long time can control GVHD but then break through. And in fact, the ones that cause what we call hyperacute GVHD are either that no prophylaxis or really inadequate monotherapy prophylaxis. And then, as I said, we can also model standard of care, which really controls GVHD just as it does in patients until it starts to break through. And then some novel approaches. This is abatacept plus sirolimus. So we can really now start to ask questions about what causes hyperacute GVHD, what causes breakthrough GVHD, and how they can be distinguished from each other. And so to do that, again, we've taken the approach of doing transcriptomics. And so what we do is either at timed points during the transplant or at the time of terminal analysis, we sort CD3 cells, we sort them from the blood and many target organs, and ask what their sort of complex programming is. And so one of the first things you do when you do transcriptomic analysis, of course, is you do principal component analysis. And one of the things we wanted to ask at first was there a difference transcriptomically between this hyperacute GVHD and the breakthrough GVHD? Did the T cells that cause these very different diseases look different from an immune pathway standpoint? And indeed they did. So this is the transcriptomics. This is principal component 1, principal component 2. And you can see that there is clear separation between those T cells in the hyperacute animals and the breakthrough animals. So this suggested that we're going to find some pathway differences as well. So we went on to look at that. So what we found, and what our goal was to really identify the pathways that, that characterize these different phases of GVHD, we first looked at the hyperacute GVHD, and what we find is that hyperacute GVHD is dominated by these highly proliferative Th1, Tc1 responses. So here's flow cytometry there of Ki67 proliferation marker. You can see that they're highly Ki67 positive in the hyperacute GVHD. And as we're all thinking about CAR T therapies here, we also know that CAR T therapies, when they first enter the patient, are very highly proliferative. There may be some very important similarities between the programming here and the programming in CAR T cell therapy. So not only were they hyper proliferative, but they also demonstrate a Th1 type immune profile. So when we look at individual transcripts, we see upregulation of some canonical Th1 transcripts, IL-12, TBET, CCR5, CXCR3. And then when we do gene set enrichment analysis, you can see there's high enrichment in the hyperacute recipients in the Th1 versus Th17 gene lists. So that information is very interesting. It's relevant to a lot of our T-cell adaptive immunotherapies. It's also relevant to the post-transplant cytoxin model of haploidentical transplant, because remember, in that model, you don't give any GVHD prophylaxis until after transplant, and we do see patients that break through with GVHD that looks very much like this. But really, the majority of our patients are not given no prophylaxis. In fact, they're given very effective prophylaxis, at least early after transplant. But eventually they break through, and so we really want to understand what the mechanisms driving this breakthrough GVHD were. And remember, these are T cells that are basically seeing alloantigen for a long time. What happens when they fail to be controlled ultimately by the immunosuppression, either the standard or novel immunosuppression? And so what we've been able to do, and as I published last year in Blood, is really look at the transcriptomic hallmarks of breakthrough GVHD as well. And we find while hyperacute GVHD is very easy to characterize, all the T cells are sort of lockstep, hyperproliferative, and very Th1, the breakthrough GVHD is actually a lot more complex and actually has 3 key characteristics. So the first thing, this was really interesting, is actually while hyperacute was very proliferative, breakthrough GVHD, so this is when T cells that are seeing alloantigen for a long time, really show a lack of proliferation and a lack of canonical markers of cytotoxicity. So here's the 2 canonical markers. There's Ki67. These are transcripts in granzyme B, and you can see hyperacute is very highly upregulated for both. But really surprisingly, not only when the GVHD is suppressed, but also when it's breaking through, these T cells are not proliferative. And it was quite a surprising observation for us. So you have pathology, you have T cells, but they're not proliferating. Why are they causing disease? And what we found was that while they're not proliferating, they're not dying either. So there's really an inhibition of apoptosis in these T cells that are sort of tonically activated. So to do that, to really delve into this, we thought the best comparison is not breakthrough versus hyperacute, but actually asking if you take time points of the same animals when the GVHD is suppressed, how do the T cells differ at that point than when they're breaking through with GVHD? And what that allowed us to see was this really profound impact on apoptosis. So there were only 12 genes when you use a very strict cutoff of statistical significance that were different. Remember, they're the same animals, so the T cells are very similar. And fully 6 of them drove towards an inhibition of apoptosis. So 3 anti-apoptotic genes were upregulated, 3 pro-apoptotic genes were downregulated, sending us a message that while these T cells aren't proliferating, they're also not dying. And then finally, we asked what are the pathways that are upregulated in these tonically activated T cells. And what we see is a complex program of both inflammation and TH and TC17 programming. So if you look at individual transcripts, you see upregulation of canonical TH17 markers, IL-17, IL-22, ROR-C, et cetera. If you look flow cytometrically, you see that in breakthrough T cells, you get more IL-17, both in CD4s and CD8s compared to the hyperacute. And then if you do fully unsupervised analysis, WGCNA analysis, which we're able to do in this model because we really can get enough numbers to really do this unsupervised analysis, what you see is these very strong networks coming through with the breakthrough T cells that really center around ROR-C, the TH17 master regulator, and then have a number of TH17-mediated genes, but also inflammatory genes. And so the insights that we've gained from the GVHD model are as follows as they relate to our next chore, which is to develop a model of CAR T cell therapy. So what we found from the GVHD model is the following. First of all, despite the complexity, we have been able to create a non-human primate model of GVHD. It closely recapitulates human disease, both clinical and pathological. We've been able to obtain detailed insight into the mechanisms of immune dysfunction using this model. And then we're able also, really tied to this, to test human prevention and treatment modalities that are being used in the clinic or that we want to send to the clinic. What we find is that early GVHD is driven by proliferation, cytotoxicity, and TH and TC1 predominant T cell activation, and breakthrough GVHD in that tonically activated state, which is an ongoing problem in the clinic with both standard and novel immunomodulation, is driven by T cell survival, so not proliferation, and this inflammatory TH and TC17 mediated activation program. And so what we ask next as we continue to study this model and really look towards using this model to get novel therapies in the clinic, we became very interested in whether we could also use the non-human primates and these insights that we've gained from the T cell dysfunction in GVHD to point us towards a non-human primate model of CAR T cell therapy. And could this model actually inform our thinking about the toxicities associated with CAR Ts? And remember, this was really inspired by the fact that, somewhat surprisingly, we found that the brain is also a target organ of GVHD. So we already knew we could model T cell infiltration into the brain. We had lots of tools to sort of deeply study them, and this made us think that we may be able to create a non-human primate model of neurotoxicity. So I'll return now to our problem. We've talked about what our lab is doing to understand the mechanisms driving one of the most important toxicities of transplant. But remember, in all those patients that we transplant, the first problem that we always face, and everybody who does this clinically knows this, is relapse of primary disease. It's really imperative in our field that we find novel therapies either before transplant or after transplant to help prevent the relapse that we unfortunately see in so many of our patients. And as we all know, CAR T cells have emerged as a really disruptive technology able to induce remissions even in the most refractory patients, and was named the breakthrough of the year just several years ago in 2013. However, we also know that cytokine release, neurotoxicity, and cerebral edema remain unpredictable, treatment-resistant, and life-threatening toxicities. In fact, this is work from Rebecca Gardner at Seattle Children's, which shows in the trials that we are performing at Seattle Children's that Rebecca leads that, and this recapitulates data from around the country. This is neurotoxicity in the CD19 CAR T-cell therapy model and the trial known as PLAT2, that fully half of our patients have some degree of neurotoxicity, and for many of them it's life-threatening. So this remains a really important problem. So the challenge that we decided to take on was, can we create a non-human primate of CAR T-cell efficacy and toxicity to uncover mechanisms and drive therapeutic discovery? The reason that we thought the non-human primate model was important for this is, again, until now, immunologically relevant animal models are lacking in the field. We need them to reproduce the spectrum of toxicity observed in the CD19 clinical trials if we're really going to make progress here. And as I said before, the ideal model would allow for testing of clinical applicable therapeutics to either ameliorate or prevent the severe side effects. So we thought the non-human primate might be a good model for this. So we set about developing this model. And as I said, one of the advantages of the primate model is that it closely recapitulates the human immune system. We have the availability of reagents, vectors, etc. There was one problem that stymied the field for many years. Of all the antibodies to not be cross-reactive, and I'll tell you, we use human panels all the time, human CD19 does not cross-react. I think there's one antibody, but it's not very good. So human anti-CD19 antibodies do not cross-react with rhesus 19, and this really stymied us for a long time. But the good news is, and we're learning more and more as we develop CD22 CARs, et cetera, that anti-CD20 from humans does cross-react with non-human primates. And so in very close collaboration with Mike Jensen's lab, who actually created this vector, we designed an anti-CD20 CAR that, again, does cross-react with rhesus CD20. So here's a CD20 SCFV. It's a second-generation CAR. We use a 4-1BB signaling domain, the CD28 transmembrane domain, and we have a marker, the EGFR-T, so we can track our CAR T cells. The manufacturing and in vitro activity of this CAR was very successful, so we do a very simple manufacturing paradigm where we start culturing the cells at day 0. We transduce with our CAR construct on day 2 or 3. We remove beads at day 6 or 7, and we do flow at day 7 to 14 to look at our transduction efficiency. And we usually get very good transduction efficiency. Here's about 68% in this example. And just as in human CAR T cells, the cultures are usually about 60% CD8, 30% CD4, and we don't do a selected product. We give everything that we transduce. The CD20 CARs can produce cytokines when given the right stimulus, and also are cytotoxic, so really had really nice in vitro activity. And so what we decided to do is, just as we did in the GVHD model, to develop a clinical paradigm for tracking these cells and seeing what they did. So we made an adaptive transfer of these CARs, and what we used is Cytoxan lymphodepletion. So as I said, one of the nice advantages of the primate is we can do all the chemotherapies at the same doses as we give patients. So in this paradigm, we gave Cytoxan. Eventually, we just used 40 mg per kg on days -7 to -3. We gave CAR T cells at day 0, and then we did some very intensive monitoring, both clinical monitoring for neurotoxicities, as well as collection from the blood, the CSF, and lymph nodes and bone marrow, and then all organs at the time of terminal analysis. As I said, sort of one of the most important things that we did in this model was really do close monitoring for neurologic symptoms. And in fact, we didn't really have a good scale to use for this, so we had to develop one. And you can see that what we monitor in these animals, we monitor them twice a day. We monitor their activity level, their movement, how well they're moving their extremities, whether they had tremor, how their gait was, normal or ataxic, and whether they had seizure. So I'll show you the data now for what we find when we give these T cells to the animals. So the first thing we saw was that indeed CD20 CAR T cells were able to expand in the monkeys, and they could induce B-cell aplasia. So again, this is the expansion of those cells. So I'm showing you 4 animals that we've done this experiment on now. And really, I want to highlight a very important control. When we started this work, we did a non-CAR T GFP control, and you can see that when you put that non-antigen stimulated CAR in the animals, you get no expansion. However, when you put the CAR20 construct in, you get really good expansion in all of the animals that we've tested so far. And just as you see in humans, as we hope to see in the primates, again, the GFP does not cause any B-cell aplasia, but you get really profound B-cell aplasia when you put the CD20 CAR in. So it really checks the box for efficacy in terms of both expansion and the on-target effect of B-cell aplasia. So as I said, we monitor these cells very carefully, and I'll tell you some of the characteristics of the expanding cells that we see in the primate. The first thing we see, and this is very similar to what we see in humans, is that the expanding cells demonstrate a CD8 predominant phenotype. So you can see this very well in R302 here that went out a long term, where you have predominance of CD8 cells after transplant compared to CD4s. Again, just similar to what we see in patients, these cells take on a central memory phenotype defined in primates as double positive or CD28 and CD95 after transplant. Importantly, this happens both in the CD4 cells that are often central memory, but also in the CD8 cells after transplant. And one of the surprising things, and actually if you look at the human data, it's there, but I don't think it's been sort of expanded on fully, is that we do not only see expansion of CAR T cells, we also see expansion of CAR negative T cells. And I'll show you this here. This is really nicely demonstrated for R302, where you see the first wave is the proliferation of the CAR T cells, but then surprisingly you see a second wave of proliferation of CAR negative T cells, very likely from the huge cytokine storm that happens that's going to induce bystander proliferation of these CAR negative cells. And indeed what you can see is that if you look at the percentage of CD3s of the CAR positive and CAR negative T cells, there's a small window where the CAR positives actually predominate, but usually in these animals, the CAR negative T cells still predominate. Finally, the third characteristic that we see in these, or the final characteristic that we've seen in these animals is, I think, very important when we think about therapeutics, is these T cells express multiple integrins, including alpha-4 beta-1. So here I'm showing you flow cytometry for an animal where we can look at CAR negative and CAR positive cells. We can subpopulate between CD4s and CD8s, and we can look again at those CAR positive and CAR negative cells, and you can see that many of them express the integrins, and this could be very important, as we all know that there are targeted therapies to inhibit this integrin, namely the natalizumab, which is approved for MS. So I've shown you that the CAR T cells expand. I've shown you that they cause B-cell aplasia. The next thing, of course, we were very interested in was whether they induce clinical and immunological cytokine release, and indeed they do. So we can look at all the clinical factors we see in patients. We can look at CRP, LDH, and ferritin. All of those increase at the same pace as CAR expansion. And then we can also look at key cytokines in the blood. And again, this is one of the canonical cytokines, of course, from humans, IL-6. But we also see increases in IL-8 and IL-1 receptor antagonists, amongst others. So we really have a nice recapitulation of what we see in human disease. And then, of course, one of the important things that we wanted to look at here were the neurologic side effects. So I'll remind you again of the grading scale that we developed. And what we can see in these animals, and it's very clear in each of the animals, is that, again, just at the peak of CAR T cell expansion, these animals start to have neurologic side effects. One of the key side effects that they have is they all develop tremor. Because of, we don't want the animals to be uncomfortable at all, we give them Keppra as soon as they start tremor, so we don't see the natural history of the disease here, but we can document significant tremor. And the other really important thing that they show is ataxia. So we really have very distinct neurological side effects, behavioral, that we can track in these animals. The fact that they develop neurological side effects obviously suggests that they are going to have abnormalities in both their CSF and their brain, and this is indeed what we saw. So one of the things that we can do longitudinally, of course, in these animals is look at their CSF. And when we do that, again, we see something very interesting. We see accumulation, again, both of CAR positive, tracked by EGFR-T, but also CAR negative cells in the CSF. And in fact, the CAR accumulation in the CSF looks very similar to what we see in the peripheral blood, both in the numbers and the proportion of CAR T cells and in the CD8, CD4 balance, which favors CD8 cells both in the peripheral blood and in the CSF in these animals. interestingly, while the cellular infiltrate into the CSF really mirrors the peripheral blood, one of the things that we picked up here, and there's been some indication that this also occurs in patients, is that there's selected cytokines that actually accumulate in the CSF. And I think that's going to be very important as we think about how we're going to both track neurotoxicity and prevent or treat it. So if you look at, this is from a single animal where you see the blood cytokine profile here, and if you look at the CSF, we're getting significantly more cytokines in the CSF than in the blood. So really interesting and really suggests we have a very multimodal inflammatory milieu going on. And finally, one of the strengths of the primate models, we can look at the brain as well, and of course that's what we're most interested in. And what we see also, not surprisingly, given what we're seeing in the CSF, is these CAR T cells also get in the brain and they cause pathology. So here's a terminal experiment we did at day 8 post-CAR T cell transfer, where you see again, at this time in the peripheral blood, fully 80% of the T cells are CAR T cells. And when we look in the brain, and we've looked at many different areas of the brain, I'll show you some of that earlier, we don't see any predominance in any area we've looked at. This is sort of whole brain here. Again, when we look at the T cells, we do a T cell prep from the brain, and it really closely mirrors what we're seeing in the peripheral blood. These animals have been fully, we do a very careful washout of all the peripheral blood of these animals before we do our organ dissection, and we know that we have confidence that this is not peripheral blood contamination here. And indeed, when we look histopathologically, we can see significant T cell infiltration in these in the brains of these animals. And really importantly, given the fact that edema is what's very closely associated with the cell deaths in CAR T cell therapy, we actually see signs of vascular edema in these animals as well. Indeed, I told you that this work in some ways was prompted by our observation that when we look at GVHD, we see central nervous system brain infiltration of T cells in GVHD. And what's really striking here is that, now here's the GVHD, this is where we've done some quantification of different segments of the brain, cerebellum, parietal lobe, thalamus, counting T cells. And this is significant. This is a lot of T cells in the brain, but look what we see in the CAR T cell animals. It's really striking how many T cells are getting to the brain in this highly inflammatory environment. And finally, what we really want to know, does this model really recapitulate what we're seeing histopathologically in the brain? And really what sort of drives home that this may be a very good model for modeling neurotoxicity is that this is an example of immunofluorescence showing CD3 in green, SMA, so blood vessel marker in red, and DAPI nuclei in blue, showing the perivascular accumulation of CAR T cells in the non-human primate model. And this is actually a brain slice from one of our patients that died of cerebral edema, again showing very similar pathology, suggesting that we really may have a way to model this deadly complication of CAR T cells. So the conclusions are as follows. Our experience with non-human primate GVHD models has allowed us to develop a non-human primate model of CAR T cell efficacy and neurotox. We see that it recapitulates CAR T cell expansion. It's associated with proliferation and expansion of both CAR-positive and CAR-negative cells, and I think that's really an important point. We have a highly inflammatory milieu here that is not just going to be antigen-specific in any case. We know that there are not CD19 and CD20 antigens in the brain, so it's not surprising that the brain infiltration is not antigen-specific, but it's also not CAR T-cell specific. We get the CAR-negative cells. We see that our CAR T-cell expansion recapitulates B-cell aplasia, also the important clinical syndromes of cytokine release and clinical neurotoxicity. As I said, we see marked T-cell infiltration into both the CSF and the brain, and in fact, the brain infiltration is greater even than what we see in untreated GVHD. Again, there's a pancerebritis in the brain as well, CAR-positive and CAR-negative T-cells. Importantly, there's accumulation of cytokines in the CSF compared to the serum. These T cells express multiple integrins, including clinically important integrins, including alpha-4 beta-1, and the cerebral pathology is very similar to human pathology. And so in closing, these results suggest that it's a very complex program of intrinsic and bystander T cell activation that is associated with neurotoxicity, associated with both soluble and cell-specific mediators of inflammation. And really, these begin to identify multiple potential targeted pathways, but I think actually the complexity means that we'll probably have to tackle multiple pathways, which can now be rigorously interpreted with the non-human primate model. And in closing, I'll thank my lab, Agna Tarasevic-Chute, who's a fellow in the lab, was a driver of this research. This was all done in really close collaboration with the Jensen Lab and funding by Juno Therapeutics with close collaboration of Raphael Ponce as well, and really important contributions by Denny Liggett and Jessica Snyder of the UW Department of Comparative Medicine. And with that, I'll thank you for your attention.
Thank you very much. We are a little bit late. We have time for a quick question. If somebody has a question, can come here in front, please.
Yeah, so that was beautiful work there. I'm just curious if you've done any comparative studies in vitro or the cells that expanded in vivo between your rhesus T cells and what we know what happens with human T cells, if there's any behavior differences between the two. I was struck by the alpha-4 beta-1 integrin upregulation. I'm kind of curious if that compares well with human.
So we haven't done that. I believe that people are starting to look at alpha-4 beta-1 in the human T cells. I wouldn't be surprised if it's upregulated because it's really an effector marker, and we haven't seen anything that really is unique in terms of effector in these T cells. We're doing transcriptomics now to look at that, but it's a very important point because obviously if we try to do targeted prevention, which is obviously where we're going, we want to make sure that those are parallel pathways that we can monitor.
Great, thank you.
Okay, quick.
Very nice.
Thank you.
Murad Hassani from Office of Science Policy at NIH. Mm-hmm. We had a workshop on cytokine release syndrome 2 years ago, and Dr. Nath from NIH, a neurologist, pointed out that there is evidence in some cases that CD19 expression in the brain tissue has been seen. So I want to just point that out. The other thing is, in our hand, I review all of the severe adverse events on these trials. We do not see the type and the extent of neurotoxicity that you see in other CAR trials or other immunotherapy trials that we see with CD19 CARs. Like, we do see CRS in BCMA CARs with multiple myeloma, but the neurotoxicity that is seen with CAR trials, it seems to be much higher with CD19 CARs. In addition, the brain edema cases have been mostly focused on certain populations, mostly ALL patients.
Can you short your question? Sorry.
Yeah, so I'll just say that one of the by using the CD20 CAR, for me it strengthens the idea that this is very likely not an antigen-specific, antigen-driven process in the humans, because it would be very odd that you'd see. We've done controls, I haven't shown them up here, where we can't see CD19 in the non-human primate brains. Obviously, we haven't done all the studies in humans, but we also don't see CD20. So that's much lower on my differential in terms of what's causing this in the primates. And then your question about the cytokine release and the severity of neurotox, my working hypothesis is this about antigen load. So even though we don't have tumor in this model, we have a lot of B cells, and I think that's probably what's causing this just overarching, huge inflammatory milieu to develop.
Thank you very much. Thank you. Okay, now as a last speaker, we invite James Wilson with the AAV-associated hepatotoxicity. Sorry. Thank you very much.
Can we go to the next presentation? Great, thank you. Renamed the talk Immunology of AAV liver gene therapy, but it was really precipitated by the concern of hepatotoxicity in many of the AAV in vivo gene therapy studies. These are my disclosures of conflicts of interest, various companies I founded and I serve as an advisor for. I think the reason I was asked to give this talk related to the bulk of experience that has emerged in the field over a number of years following in vivo AAV gene therapy. And a number of diseases require a systemic delivery of vector. I've just summarized here ongoing hemophilia B studies, try to get a sense of what context are we considering this discussion today. These are 4 different studies. 4 different capsids. The lead was the work out at St. Jude's and UCL, really pioneering work which showed the ability to achieve stable expression of factor IX following in vivo delivery of gene therapy. However, there was associated in a number of these research subjects a transient, sometimes not out of the range of normal, elevation in transaminase, which appeared to be of more concern because it was correlated with a drop of transgene expression. Well, since that early work, a number of other groups have moved forward, Dimension, Spark, Unicure. And if you take a step back and try to assess what has the experience been, there is consistently, independent of manufacturing capsid, et cetera, observations of transient elevations of transaminases, and I count about 40% this is observed. So I guess what I've been asked today is to try to explain why it occurs and how we're going to prevent it. I wish I had those answers, but I do want to share with you all unpublished data, clinical and preclinical, that maybe starts to define what the issues are and how we can go forward. So before I get into the data, I just want to provide a baseline level of background about immune problems. The immune problems we're talking about potentially are the generation of killer T cells or cytotoxic T lymphocytes that are CD8. And as this story has emerged, we've considered a number of different targets to which these T cells are directed. The classic approach would be a vector transduces an antigen-presenting cell, and the transgene product, or a product of the transgene, maybe a little different, is presented by class 1 to activate CD8 T cells that then can target other antigen-expressing cells, such as hepatocytes. There's a requirement for helper T cells, so the capsids usually derive those responses that are class 2 restricted and in a way activate the CD8 T cells. So a hypothesis that emerged from work out of CHOP and Stanford early on was the notion that maybe the antigenic stimulus for the killer T cells was not a classic MHC class 1 response, but was a response due to the cross-presentation of capsid antigens, and this led to the capsid T cell hypothesis, meaning that our problem is really the fact that the hepatocytes take up the capsid, present antigen, and are targeted by T cells that are activated. So in the backdrop of this, there's another story that I believe is an incredibly important story, which has to do with the observation that this vector system, unlike any other system I've worked in, has developed or exists a counter-regulatory strategy to actually suppress immune responses. So those are what are called Tregs. And basic viral vectors usually activate the effector CD8s, and you get this kind of immune toxicity, or if it's a vaccine, you get efficacy. But there's something different about AAV in that it has the ability in a transgene-specific way to actually activate Tregs, so there's this balance. And as we traverse the data, both what I'm going to describe today and in the literature and go forward, we have to be cognizant of the fact that maybe what we're actually studying is the balance of the Tregs versus the T effectors. Tregs are hard to identify, to study. T effectors are easier to do so. So I'm going to describe to you 2 new data sets that haven't been published and haven't been presented. One in a clinical trial that we've been involved in, and another in a pretty interesting non-human primate study that hopefully will start to bring together some themes. So the trial, the study that we're going to talk about today in the first half of my talk is the clinical trial of hemophilia B that our colleagues at Penn have been collaborating with Dimension. And this is a phase 1 dose escalation study using AAV-RH10, which is in the same general category of vectors such as AAV8, but different, expressing factor IX from a stranded genome. And the primary endpoints were as safety primarily, and then factor IX activity. Patients were screened to be low in pre-existing antibodies to AAV-RH10. The plan was to traverse through 3 doses in a Bayesian type approach, and the study has progressed through cohorts 1 and 2, and the company provided an interim analysis of this around early March, describing the transaminases in the level of factor 9 expression in these 6 patients. And I don't have any more data beyond that to share with you today, but the company will be presenting that in a subsequent scientific meeting with the PIs that have been involved in the study. I'll briefly summarize what was provided at the end of March, but then describe our thorough kind of analysis of the immunology of this trial. So again, this was a part of the disclosure early March, summarizing the 6 subjects, 2 doses, low dose, and then the next dose, their ages. Basically, what was observed in the first 5 subjects was not that different, wasn't different qualitatively that one had been described in the previous studies, that there was a transient elevation in ALT that seemed to be coming down, one that wasn't really significant, and then reasonable levels of peak factor 9 expression in which there was some diminution at the time that the study was reported out. But then in subject 6, there was a qualitatively different response. In this subject, 48-year-old, who otherwise was healthy and normal and qualified for the study, realized a fairly quick but high elevation in transaminase to over 900 that at the time with treatment with steroids started to decrease. And obviously this kind of response deserves attention. The study was stopped, and we came together to try to do a comprehensive analysis of the immunology of this experiment and what was different about this subject versus the others. And that's what I'm going to share with you today. So the first thing is, how about the antibody response to the capsid? Now that's not probably causative to any toxicity or any effect on the transgene-containing cells. But it would be some indication of immune memory. And so this is the 6-week level of NAB against the AV capsid, and this is 1280, a little bit higher here, and typical of what we've seen. But what was striking was there was a very quick and rapid increase in NAB to the capsid in the 6th subject. So this was about 10-fold higher or even greater than what was seen in the previous subjects. Again, that's not causative, but it would suggest there may be some level of immune memory or pre-activated T cells or activated immune status in this subject. So then bear with me, the so-called LA spots that we all have to wade ourselves through. So this is a very rudimentary way to measure from any tissue, in this particular case, peripheral blood, for the presence of T cells that are activated to either the capsid or the transgene product, and that would be factor 9, both of which could be considered antigens. And so let me just walk you through the data. These are the 6 subjects, and what we have here is the LA spot readout, which is spot-forming units, over time. And in each case, there's a control, which is the first bar, and then there's a series of green, which are pools of peptides against the capsid, and then in color, pools of peptides against factor 9. And you'll see that this kind of goes up and down. A positive result is indicated by an asterisk, and it's required that the signal is several fold above the baseline, and baselines vary. So this isn't a positive because the baseline is high. So if you just focus on the indication, the stars here, that in the first 2 subjects there's no evidence of activated T cells by expressing interferon to capsid or transgene. And in the subject 3, and we'll talk about subject 3 in a minute for other reasons, there was by 6 weeks T cells against the capsid, but they're very low. I mean, remember these numbers, this is about 60, and then they went down. And then the high-dose cohort, again, the 6th subject is the one that's the focus of our attention. We did get pretty significant activation of capsid T cells, but not factor 9 in the blood here in subject 4 and 5 that were higher, so it's dose-dependent. But the one that we obviously were really anxious to evaluate was subject 6, and that was a very weak, barely significant elevation of ELISpots against factor 9 or capsid in that subject. How could that be? What's causing the transaminase elevation? Well, let's continue to explore that. Another interesting fingerprint of an immune response is simply to take the serum and measure in a multiplex way every conceivable cytokine that you can. And what we have here is a heat map of levels of all these various cytokines and chemokines in subjects 1, 2, 3, 4, 5, 6, and this is over time. So if we want to see if there's a response of the subject to the vector, let's see if there's a change over these intervals. And the one that really initially stood out to me was subject 3, in which there seems to be sort of an escalation of activation of cytokines and chemokines. That was the one that had the highest level of the gamma T cells, but no clinical sequelae apparently as a result of this, and it rapidly then came down. But let's look at subject 6, which really seems modest. But what was interesting about 6 is it's the only subject in which there's sort of a pattern of a Treg. So we have TNF alpha, we have IL-10 and IL-7. That's very different from the others. So again, let's just check that. From a cytokine standpoint, not a real active inflammatory response, but what may appear to be a unique Treg response. And then finally, I happen to believe that one very important parameter about how our recipients or patients are going to respond to gene therapy is their HLA genotype. And so we asked the question based on the HLA typing of the 6 subjects, which we did type, using computer modeling. Do any of them have high affinity binding to peptides that span their mutations? And this isn't proof, but it would be suggestive that possibly this could be related to a T cell response against the transgene product. And what we're looking for here, for example, in this case, these are the different HLA types. This is a mutation queried against the computer modeling. And the high number here is that peptide was actually quite low on the hierarchy, whereas when the number is higher, it was higher on the hierarchy. And what stood out to us was subject 6, that's sort of independent of the 4 different alleles that we were able to characterize, that they all ranked high in predicted binding to the peptide that spanned that mutation. So at this point, those are the data. We are, again, the company and the PIs are going to report at a subsequent meeting the ongoing clinical data. But what have we learned about this study? Quite frankly, what may be remarkable about what we learned is how unremarkable the data are in subject 6, who had a very high level of transaminase. And suffice it to say that the clinical team has ruled out any other reasons why that patient may have had that transaminase. But there are some clues. So in subjects 1 through 5, there was really nothing different from what had been observed in the other trials. Transient elevations of transaminases, T cell responses to capsid, very low, 50 to 100 or so, and not clearly related, although you can't really determine causation. But subject 6 was different because of the high and early elevation in the transaminases, but there was no clear etiology, as I indicated, unrelated to vector or related to vector other than temporal. But there was a different response to vector in the subject. First of all, there was this high, very early NAB spike, suggesting immune memory. And the serum cytokine profile consistent with exaggerated Tregs. And when, in my life in studying adenoviruses and responses to adenoviruses, when you see a vibrant Treg response, it's in response to a T effector response. It's almost the consequences of. And that would suggest maybe that there was some T effector response that we didn't detect. And that this mutation in factor 9 and HLA class is a hotspot. So in the past, we've said, look, if there are no ELISpot positives to the transgene product is absolved. And maybe that's sort of what we would have concluded here. But I don't think it's that simple. So I'd like to share with you a study that we've done in collaboration with Audentis. And it was part of a non-clinical study in preparation for a clinical trial treating patients with AV8 with Crigler-Najjar type 1 hyperbilirubinemia study. And in this study, there were male and female macaques. The route of administration was intravenous. Again, different capsid from that previous study, but still inexperienced with AAV. There was a control vehicle group with 2 animals, a dose at 10 to the 13th genome copies per kilo for 3, and then a higher dose, 2.5 times 10 to the 13th. So these are highish doses. The time points, we did a liver biopsy at day 14, a necropsy at day 56, and T-cell assays at 0, 28, and 56, and then the animals were necropsy. So I'm going to show you the low dose, the high dose, and then the control or vehicle control. Again, a complicated slide, but let's just walk through it. What we have here is time, vector, biopsy, necropsy, blood, transaminases, ALT, AST here, and then again this kind of LE spot motif where there's the no stem, then there's the capsid, and then the transgene. If you focus on wherever there's a star, we have a signal above background. Now, the difference between this study with respect to analysis and anything you could do in a human is we can get tissue at necropsy and we can extract lymphocytes from those tissues and determine whether there's evidence at the site where the vector went of immune activation. So we have liver, spleen, and bone marrow here. The first animal, we had pretty significant T cells against the transgene product. This is a human version of the enzyme in a macaque. Very little to capsid, maybe very little, but definitely we had capsid transgene responses and a transient increase in transaminases that wasn't really high, but was detectable. These animals did not get steroids or any immune modulation, self-limited. But what we also then saw in tissues, in particular liver in this animal, was very high T cells against the transgene product. So T cells in the blood, T cells in the liver, transient transaminase elevation. Second animal, somewhat of a delayed transaminitis, still significant. Very high T cells in the liver, nothing in the blood. So if we would have just measured blood, we would have missed the fact that this animal had high levels of transgene-specific T cells. This was a really odd animal that had pre-existing T cells against the transgene product. And that's been an observation in Duchenne's and others. And that animal really was uneventful in terms of transaminases and also any T cell responses in tissue. Let's go to the high dose. And what do we see? Same motif, transaminases here, up. Again, not super high. Start to come down. T cells against the transgene product in blood. And again, very high level of T cells, actually higher in bone marrow, spleen, and liver. Second animal, elevations of transaminases, very little, if any, in the blood and very high in the tissue. Again, another example where sampling blood, we would have missed it. And then the third animal, we had elevated transaminases and some T cells very low in the blood and background in the tissues. Now, these are controls that receive vehicle and really super clean with transaminases with respect to T cells. These are elevated because of the background, but nothing above background. So how about transgene expression in these animals? This is an in situ hybridization of an animal that received the high, the low, and then the vehicle, showing pretty high levels of transduction. This is relative to area, so you've got to multiply it by at least 2 for hepatocytes. These are the high, low, and then the control. And how about effect on expression? So we actually had a biopsy at day 14 that we could do PCR, DNA and RNA, and then at 2 months. And these are paired samples here, before a biopsy and then later. This is DNA and this is RNA. And really to the animal there was some reduction, but in many cases it was about 3 to 4 to 5-fold and sometimes less. And it's interesting, the 2 animals that that had the smallest, lowest transaminase elevation had the least drop in transgene expression. So what's going on, and can we learn more about this response? Well, we had a T cell response against the transgene product, and it was a human protein in a macaque. So how did that happen? Well, this is an amino acid comparison of human and macaque, UGT1A1, which is the protein involved. And wherever there's a shaded area, there's a conservative change, and a white area, a non-conservative change. So the proteins differed by less than 5%, and I often hear when we think about experiments, well, the human only differs by a few percent. It shouldn't be a problem. Well, this was only less than a few percent, and it was a problem. And so we were able to actually map the T cell epitope to which the T cells were directed. And it was, because the frequencies were so high, it was here, spanning this epitope, this peptide here, in which there's an isoleucine versus a methionine that may be conservative, which was the target of the T cells in all the animals. And this should be class 1 restricted, and there should be some variation, and we're still analyzing HLA in the animals. So anyway, despite the fact that there was a lot of conservation, just one amino acid difference in the wrong place of the protein, which happened probably to be highly presented by class 1, we got a pretty significant transgene response. Now, what about the T cells that ensued? So ELISpot measures one activation marker, gamma interferon, and you can do more sophisticated studies using multicolor flow, which we did, in which we analyzed a variety of memory markers, functional markers, and trafficking of homing. Let's look at the functional markers. So the way this works is you take the T cells out, you give them antigen, study them before and after antigen to see if they're activated. And what we're going to focus on is whether there's evidence for cytolytic activity. The two most important markers are granzyme, which are a functional enzyme that's released from CTL, and then CD107A, that's kind of the remnants of that release that's found on the surface. So I want you to just to remember CD107A, because this has become very important. So when there has been active release of granules, you get high levels of CD107A in proportion to how many cells were activated. So this is a simple view of the activation of those markers from spleen of one of the animals. And this is recapitulated throughout all of the tissue analyses. And what we have here are CD8 cells in which we then stimulate with peptides to the protein. Don't stimulate or stimulate. And so what we're looking for, is there a difference in these two buckets? First of all, granzyme is high. There's a mixture of high and low not affected by stimulation. And there really is very little evidence for activation of cytokines if you compare here to here, maybe modest in some animals. But we had a positive gamma ELISpot, so there are activated T cells. But what was actually remarkable was the activation of CD107A. Over 50% of the cells in response to antigen, you can see here, unlike any response that our human immunologists had ever seen, suggesting that there was some massive sort of activation of these cells, or at least presence of these cells in the liver. Now, we activate them in vitro. That doesn't mean that they were necessarily activated in vivo. But it's a very high frequency in the liver. And that's an abnormal phenotype, because usually with release of, or the presence of 107A, you get cytokines that are upregulated as well. So let's stick with this theme. Does the blood reflect the liver? Well, clearly there are examples where there are no T cells in the blood and a lot of T cells in the liver. How about this phenotype of high CD107A? In animals in which there are T cells in the blood, is it the same in the liver? And this is an example of an animal, high T cells in liver, high T cells in blood, really. And then what we've studied here are the no stimulation controls, and then the stimulation controls. So what we're really looking at is, does this go up? Does this go up? Does this go up? And this is CD107A and granzyme. So As I showed before, CD107A goes up significantly with antigen in spleen and liver, but nothing in blood. So blood not only doesn't reflect the quantity of T cells, it doesn't reflect the quality of T cells that are activated in response to antigen. And then one last point, and then we'll summarize and wrap up, was we actually activated a T cell against a peptide that differs by one amino acid amongst very few. And we wanted to know whether that activation to that foreign peptide may have actually activated to the self-peptide as well. So this is an experiment where we synthesized the peptide to the transgene-specific target and then to the self, that I think was at valine to methionine, and then stimulated the cells with either of those peptides. These are the no-stim controls, and this is a spleen from one animal. No stim, nothing. To the self-peptide, very little. To the transgene peptide, very high. So this was a T-cell response specific to that foreign peptide. However, there were equally a number of animals in which that T-cell activation expanded its breadth to not only activate T cells against the non-self antigen, but to the self antigen here as well. Now, are we surprised by this, that one amino acid difference can, between a transgene and a endogenous gene elicit a response? We just published a paper with our colleagues at UMass, Terry Flatt and his colleagues, where we performed a similar analysis from peripheral blood of patients that received AAV1 vector with alpha-1 antitrypsin deficiency, and we showed that one subject that showed a very robust T-cell response against the transgene product differed by only, there were 2 amino acids, and this was a polymorphism that was directed against it. In that case, it didn't seem to have clinical sequelae. So just to conclude, what did we learn about the studies in the macaques? One is that antigen-specific T cells in the blood do not reflect the quality or the quantity of the T cells in tissues. And it does give you reason to pause when evaluating our studies from the Dimension study, where in subject 6, the peripheral blood was really markedly unremarkable. The T cells directed against the UGT1A1 in tissues has this very abnormal phenotype, unlike anything seen before. Really, the granzyme doesn't go up, but the 107A does. And this suggests that, according to our group and the immunologists we work with, that while we're activating T cells, they may not be as pathogenic as they would be otherwise, probably due to this immune tolerizing nature of AAV. And it could be that their granules are void of granzyme, or there's something abnormal about it, which is fortunate because for those T cells, 50% of those T cells to potentially be responsive to antigen, if they weren't held in check, that could represent a significant safety feature. And T cells directed to a non-self peptide can break tolerance against the corresponding self peptide. So I just want to recognize the groups of people that we work with. I also want to thank my colleagues at Dimension and Audentes for allowing us to share this preliminary data that all of us felt was important to share with the community in terms of evaluating how best to proceed forward. And those in my group that did this work, Roberto, Tammy, Jenny, Maria, and Laura. Thank you very much. Happy to answer a few questions.
Thank you very much. We run out of time, so the questions will have to be asked direct. Maybe just one question.
Hi, Fraser Wright, Spark Therapeutics, Philadelphia. Thanks, Dr. Wilson, for a nice review and data presentation. 2 really quick questions. I think one of them is, can you say anything about not sure if it's been disclosed, the timing of that high ALT, I guess the 6th subject, how it seemed to be early, how early, and how tight was it? Did it come down quickly? Second, really quickly, is there's this dose response, I guess, in the ALTs, typically within any trial. Would you be willing to comment about, if you compare the various trials, Unicure, even perhaps BioMarin, where we see a very large range of the total doses, and what that might say about immunology, where one trial might be 2 logs higher than the other, but what can we learn there?
Yeah, more details on the clinical trial. I don't really have, I'm not that conversant about them, but They'll be presented at an upcoming meeting. You know, correlating transaminases with anything is difficult. Hepatologists, they're not directly related to almost anything other than they're a mark of something, for sure. All right, thank you.
Thank you very much. And so we conclude the session. Thank